my Talaria Summer Research project is on the investigation of antigen presentation after knocking in different human leukocyte antigen alleles into an MHC complex using a knockout B cell line for vaccine development. So before I get into all of that, into all the details of the research, I just want to give a little bit of the context of the whole presentation. So first, I'm going to give some background info on my mentor and I, and then I'd like to outline our experiment and a little background on immunology. And then I'll discuss the methods I've detailed and also the gene table I've created. And I will provide a conclusion and then give a big thank you. So just to start with some background information, my mentor, who is Tisha Baker, is a PhD candidate at UBC in the biochemistry department. And here she is pictured on the right, working in a lab. And so in the span of our project, she has provided the paper's foundation and given me sort of an outline. She's written up the introduction and provided general mentorship to me. As the mentee, I am currently going to enter my Bachelor of Sciences at University of Victoria. I just graduated high school, and what I've done is I summarize and discuss the methods from 3 different research articles to create a new experiment, as well as obtaining data from a database of the human leukocyte antigen cells. Okay, so I'd like to give some more general context on components of the immune system and cover some terminology before I get into the details of our experiment. So as you may know, the immune system is a complex network which protects organisms from infection, disease by identifying or eliminating foreign invaders. So components of the adaptive immune system include foreign molecules, which are known as antigens, and they can bind to a T cell. So the T cell's job is to scan and then recognize an antigen and either act directly upon the infected cell or assist in destroying the cell. So the cellular components that are responsible for presenting the antigens to the T cells are known as MHCs, or major histocompatibility complexes. All right. So the analogy I'd like to use for the antigens to help you guys understand, as that's something that my mentor helped me with when she was first explaining this all to me, was using sort of a hot dog analogy in a hot dog bun. So if you, if you look on the far right, you can see that the red dot in the MHC1 picture on the far right is basically representative of the hot dog inside the hot dog bun, which is the MHC. So the antigen is being presented on the surface of the cell over here. If you look at the number 4, you can see that it's on the cell surface. It's being expressed on the cell surface, and it's in the binding pocket, or the hot dog. So the antigen is the red dot, and the hot dog is sort of what surrounds it, right? So that's the bun, that's the MHC binding pocket. And that whole thing is what is being scanned by the T cell. So as you can see in the MHC class 1 pathway photo, you can see at the top there's a green T cell, and that's what's scanning it. And now, if you look at the bottom left, the graphics at the bottom left, another way to visualize this hot dog analogy is by looking over here. You have a top-down view, which is the first, the top photos, the E and the G, those are the top-down views. And then near the bottom are the side views of the hot dog bun and the hot dog. So what we have here is the hot dog is sitting inside the hot dog bun. And the antigen has been processed by the cell, which happens in the pathway. After it's processed, it goes to the surface. So steps 1 to 3 in this big picture over here, steps 1 to 3 are what happens when the cell, it's processing it. But what we're focusing on is step 4, which is the expression of the antigen on the cell's surface. So that's what we're focusing on for the purposes of our research, and that's what I'd like to clarify before I get into my research. Another thing is that the antigen that is being presented is also known as an immunopeptide, which is what is sitting at the surface of the cell. And this is what we use for vaccines. This is the core of vaccines. They use antigens in vaccines. And this is why vaccines, they don't make you sick, but may trick your body into believing that you are sick or you have a disease. And so that is why it produces an immune response in order to fight the disease. That's what this is all about and how this relates to vaccines. Okay, so more of an intro into my experiment. Basically, for context, around the world, there are probably 30,000 HLA genes or hot dog buns. This is what would hold the antigens, or the holder, right? And so this means we have to account for all this genetic diversity all around the world, right? Because this is just all in different geographical regions all around Earth, right? And so we only chose 6, and we chose these 6 because they are in the same cell line that the lab from the research articles that I was referring to was using. So these 6 were chosen as they are on the same cell line, and all of these different alleles have these different antigens that we want to identify on them. So these alleles come with the antigens on the surface, right? And so our goal was to identify them and observe the antigen presentation. And so in order to do this, we had to start with a knockout cell line, which I will elaborate on later. And what we were doing is we are knocking out the genes and then knocking them back in one at a time into the HLA alleles. We're knocking in the HLA alleles back in one at a time. So within this project, I detailed the methods and discussion for a brand new experiment by inserting my target genes through combining methods from several research articles. This experiment itself is yet to be done due to the time that we had here over the summer. Basically, I just detailed the methods from research articles that I read with my mentor. And so the lab itself is in process. The cells are coming over from all the way from Europe. So there's a chance that I might be on the bench when the lab itself is being done, which is very exciting. But it is a long process. So, I mean, I am just happy that I was able to detail these methods to create a new experiment. All right. And so this is more on the methods and the discussion, which I will briefly touch on. So our experimental methods are beginning with this knockout cell line. Like I mentioned before, I would elaborate on the knockout cell line. And so knockout means the gene of interest is basically you remove it, you remove the gene. And for our study, the MHC genes were knocked out so that we can knock back in one gene at a time, one HLA gene at a time. So these target HLA genes are to be chosen to match the haplotype from the cell line. Like I said, the cell line that was used in the lab. And the cell line comes from the cell line bank, the 1000 Genome Project. My genes of interest from the cell line are named HG02386. So that's the name of the cell line, very specific. And I will refer to this in the following slide. So as you can see at the top, there is a little construct. It's basically a little visual I created. And what you have is a 10 base pair primer, and then you have a gene. You have the HLA gene sequence, which is this blue one, and then you have a tail. And so what happens here is the HLA gene sequence that I have here, it represents a placeholder for my genes, the one that I found using the database, the HLA genes that we chose, those 6 genes that we chose out of the 30,000. That represents those genes because the code is very long, so I wasn't able to paste it into there. So that's a placeholder. So that is what will be transduced back into the knockout cell lines. So first it was knocked out, and now it's being knocked back in. And what's being knocked back in is this HLA gene sequence. So the last step, after we obtain the knockout cell line and we clone the genes, we transfer the HLA genes, we have to confirm the transduction of the cells by using a method called mild acid elution to identify what antigens are present on each of the different HLA alleles. So this might be a little hard to see, but in this sort of slide, I just wanted to show the HLA alleles. So what I mentioned was that the code was very long, right? And so this is a 40-second video of me scrolling through the code for each of the HLA alleles that I chose. And this is why I wouldn't put this into the construct, because the HLA gene sequence is very long. So it is these genes that are going to replace the placeholder that was illustrated in my diagram. I'll try and play it. Hopefully it'll work. Might be a bit hard to see, but that's basically a whole gene sequence. Like, it's just a bunch of little letters. It's a giant paragraph of just letters of code for the gene. And this is what I had to retrieve from the HLA database. So this is just a part of it, right? So each little bit here, this is just one code. This whole thing is one code. We're not going to watch the whole thing. It's quite long, but I'm going to move on. Let's see. Yeah, so now for my conclusion, the purpose of this experiment and sort of our objective was to have my knockout cells with their genes knocked back in one by one in order to create a new cell line which contained single HLA genes. We will then be able to see the differences in the antigens, which, as I mentioned before, these are the hot dogs. These are what are inside the bun. And how these antigens fit into all the different MHC alleles, the hot dog bun. So in this picture, you see all these different colored sort of like placeholders. These are the MHC alleles. So the MHC is holding the antigen. And in this diagram, each color is representative of a different MHC allele from all around the world. We can see, like with the colored lines, that not every hot dog fits into every bun. Accounting for this diversity is critical when we're looking at cancer immunotherapy, we're looking at vaccination and autoimmune diseases, as an antigen is, like I mentioned before, the core of a vaccine. Okay, and that is all. I just wanted to say thank you to my mentor for her amazing guidance. Thank you for the Talaria program, which I have participated in before as an officer. It's been a great experience. And thank you to BioRender, which I used to create my little construct. So yeah.